Okay, hello to all of you. Let's discuss chapter number 13. And chapter number 13 is about the another cell division, meiosis. So in chapter number 12, we have discussed about mitosis. Now we are going to discuss meiosis. And so basically before we actually, so in this chapter, I'm going to basically talk about the differences which are between mitosis and meiosis. So we are going to focus on that. And before we actually go over and talk about the main differences between these two kinds of cell divisions, we are going to go over some of the terms. So first of all, what is hereditary? Heredity is the transmission of traits from one generation to the next. Variation, it's demonstrated by the differences which you see in the population. So variation is demonstrated by differences in appearance that offspring show from parent and siblings. So in this chapter, variation basically, so variation is a broader term which means the differences between the different individuals in the population. But when we talk about the variation in the family, it means the differences the children have from their parents. And genetics is the study of hereditary and the variation. So you can see here, this is a picture of a family. This is a father, mother, and these are different children, siblings. So you can see in the family members or in the children, that some of the characteristics of the child looks like of the mother, some of the characteristics look like of the father. So that's what we are talking about in this cell division, that how do parents transmit their characters to the children, and how do some characters are same and some characters are different, or some characters are mixed of the both parents. So in this cell division in meiosis, we will understand how do all these things happen in the family. So we know that the life starts when the sperm fertilizes the egg. So basically, an individual is formed from the fertilization of the sperm with the egg. So basically, every individual is formed by the fusion of two different gametes or two sex cells, sperm from the father and egg from the mother. So basically, the genes are there, the father's genes are there in the sperm, and the mother's genes are there in the egg. And these two sex cells having the genes, they fertilize and they form one fertilized egg. And in the fertilized egg are the genes of both parents. And then other things takes place, and because of that, the individual gets the characteristic. So basically, meiosis is a cell division. Meiosis is a cell division by which sperms are formed as well as eggs are formed. So eggs are formed in the females, sperms are formed in the males. So meiosis is a cell division by which these gametes, they are formed. And in the cell division, during this cell division, which we are going to talk in detail, some events takes place, and because of which some shuffling of genes takes place, and recombinant chromosomes are formed, and that is why in the future generation we see some mixed characteristics. So we are going to talk about these things in detail. So offsprings acquire genes from parents by inheriting the chromosomes. So the chromosomes are there in the gametes. So if like we talked in chapter number 12, that in the humans we have 46 chromosomes. We have 23 pairs of chromosomes. So in the mitosis, the very, very important feature of mitosis is from one parent cell, two daughter cells are formed, and they are genetically identical to each other, which means the same kind of DNA and same amount of DNA. 46 chromosomes in the parent cell, 46 chromosomes in the daughter cells. And how does it goes from 46 to 46? Because in the interphase, there is a phase called S phase, synthesis phase, in which duplication of DNA takes place, and in the mitosis, these duplicated chromosomes, they get separated. And we also talked about the structure of the duplicated chromosome, that the duplicated chromosome has two sister chromatids, And in the anaphase of mitosis, it is the separation of the sister chromatids. So 46 chromatids go in one cell, 46 chromatids go in the another cell. And these chromatids, they represent the future chromosomes. So that is the main feature of mitosis. Now in this chapter, we are going to talk about and we are going to see how meiosis is different from mitosis. So what are the main differences, main events or main events basically take place during these stages of the meiosis, that it results in the formation of 4 daughter cells from one parent cell. So in meiosis, from one parent cell, four daughter cells are formed, and these 4 daughter cells, they are haploid. So we are going to also talk about what is diploid and what is haploid. Diploid means when there are 2 sets of chromosomes. Haploid means when there is only one set of chromosomes. Now in humans, we have 46 chromosomes, and these 46 chromosomes, they are present in 23 pairs. So which means that for every chromosome, there are 2 sets. For each chromosome has 2 sets. So in these somatic cells, we have 46 chromosomes or we have 23 pairs. So in a somatic cell, we have 46 chromosomes or we have 23 pairs, which means in every cell there are 2 sets of chromosomes. But in meiosis, from one parent cell, 4 daughter cells will be formed, and these 4 daughter cells, they are haploid, which means, so the parent cell is diploid, which means parent cell has 2 sets of chromosomes. From one diploid cell, 4 haploid cells will be formed, and haploid cells means they have 1 set of chromosomes and they will be all unique. That's what we are going to learn in this chapter, that what events takes place during this process, that it results in the formation of haploid cells from the diploid cells. So there is a reduction of number of chromosomes. And also, second very important thing is, all the cells which are formed, all the sex cells which are formed, all the gametes which are formed by the process of meiosis, they are all unique. They are all unique. So what even take place, or what happens during this process, that they all become different from each other. So this is one of the very, very important difference between mitosis and meiosis, that in mitosis, from one parent cell, 2 daughter cells are formed and they are genetically identical. In meiosis, from one parent cell, 4 daughter cells are formed. They are haploid and they are all unique. Okay, before we actually go into the phases of the meiosis and into the details of the events, we need to know some of the terms. So we already know what genes are. So genes are units of heredity and they are made up of segments of DNA. So basically, where are the genes present? The genes are present on the chromosomes. So we talked about in chapter number 12 about what is chromatin, what are chromosomes. So chromatin is a dense mass of DNA which is wrapped over proteins, and when the cell is not dividing, the DNA in the nucleus is present in the form of chromatin. When the cell starts dividing, this chromatin gets condensed and it takes the shape of chromosomes. So these genes, they are present on the chromosomes. So these genes are present on the chromosomes, and these genes are basically made of DNA wrapped over the proteins. We know what gametes are. Gametes are the sex cells, sperms in the males and eggs in the females. We know what somatic cells are. Somatic cells are all the body cells in our body, and in humans we have 46 chromosomes or we have 23 pairs. What is the locus? Gene specific position along a chromosome. So the chromosomes, the chromosomes have millions of genes on them. So every gene has a specific locus. Now we need to know what is difference between asexual and sexual reproduction. So asexual reproduction does not involve the fusion of the gametes or the sex cells, and it is basically it's a single individual passes all its genes to its offsprings. And in the sexual reproduction, there is involvement of 2 parents and there is a fusion of the 2 gametes. So this is like an animal called hydra. So basically this hydra produces one bud and this bud then separates. So this is asexual reproduction because there is no involvement of the gametes. And this is these trees, they also undergo the asexual reproduction. So basically fertilization and meiosis. So we have already talked about, I will go over here and explain you the life cycle. So basically this I talked in last chapter also. Basically so the life starts at the conception point when the sperm fertilizes the egg. When the sperm fertilizes the egg and it forms the fertilized egg, and sperm is haploid, which means it has one set of chromosome. Egg is also haploid, one set of chromosomes. So when sperm fertilizes egg, it forms a diploid zygote, and the diploid zygote has two set of chromosomes, one from the sperm, one from the egg. And then this zygote, it actually multiplies by mitosis. It multiplies by mitosis and it forms, by mitosis, there's a growth and development. It forms the adult, and when they go through puberty, then with the help of the division called meiosis, they form their own sex cells. Like males are going to produce sperms, females are going to produce eggs in the ovaries, men are going to produce eggs in the testes. So they are produced by meiosis. So they are produced by meiosis, and then again they get fertilized, and then they, so this is the life cycle. So the life is going on with the help of two kinds of cell divisions called mitosis and meiosis. So now let's see if we covered all these things. Okay. So okay. Few more important things we need to know about the chromosomes. As I said, humans, they have 23 pairs of chromosomes, and karyotype is basically the picture of chromosomes. Basically in humans we have 23 pairs of chromosomes. So in every pair there are two chromosomes, and they are called homologous chromosomes. So the two chromosomes in each pair are called homologous chromosomes. So chromosomes in a homologous pair are the same length, shape, and carry genes controlling the same inherited characters. So you can see here, this is a karyotype. Karyotype is the picture of the chromosomes, and there is a way to take the pictures. You take the cells, and then in the metaphase you take the cells which are in metaphase, you stain them, and then there's a computer attached. You can see the picture of the chromosomes. So now you can see here, this is a karyotype of humans. This is pair number 1, 2, 3, 4, 5. You can see going up to pair number pair number 22, and this is the sex chromosomes. So in every pair there are two chromosomes, and these chromosomes are the homologous chromosomes. So they are same, they have same length, same shape, and they carry the genes controlling the same inherited characteristics. So these are all, so there are 23 homologous pairs in humans. So there are, in humans, 22 pairs are called autosomes. 22 pairs are called autosomes, and one pair is called sex chromosomes. Okay, so the sex chromosomes in the females, it is XX, and in the males it's XY. So we have already talked about what is diploid, what is haploid. Okay, so basically as we talked about mitosis, in mitosis there's a duplication of DNA in the S phase, and then that duplicated DNA divides into half, and that is why from one parent cell two daughter cells are formed. They have genetically identical, they're genetically identical, and they have the same kind of DNA, they have the same amount of DNA. But in meiosis what happens is, first of all, the pairing of chromosomes takes place. So this is so in the pair, in every pair, important thing to know, in every homologous pair, one chromosome is paternal chromosome, another chromosome is maternal chromosome. One chromosome in the pair is coming from the mother, another chromosome is coming from the father. So usually in the book maternal chromosomes are shown by red color and the paternal chromosomes are shown by blue color. So first of all, in the meiosis, the pairing of chromosomes takes place. So every chromosome goes and looks for its partner, and the pairing of chromosomes takes place. And then in the meiosis, there is actually the separation of the so meiosis has two phases, meiosis 1 and meiosis 2. In meiosis 1, first of all, meiosis 1 is called reductional division because in meiosis 1, the chromosomes in the homologous pair, they separate, and that results in the reduction of the number of chromosomes. From 46, it results in the formation of 23 chromosomes. And because meiosis has two phases, meiosis 1 and meiosis 2. Meiosis 1 is preceded by interphase. Meiosis 1 is preceded by interphase, and in the interphase there is a S phase. So in the interphase there is a S phase. The DNA was duplicated there. The DNA was duplicated there. So in meiosis 1, the separation of chromosomes takes place. The number of chromosomes reduced to half. But remember that those chromosomes, they are duplicated. Those chromosomes, they are duplicated. And then in the meiosis 2, the separation of sister chromatids take place. So we are going to talk about all these things in detail now. So I have already discussed all these things, so I'm not going to repeat. Okay, so now let's talk about in detail about the cell division meiosis and the various events which takes place there. So meiosis has two phases, meiosis 1 and meiosis 2. Meiosis 1 is called reductional division because in meiosis 1, the number of chromosomes, they reduce to half. So basically this is what is happening this is the overall view of meiosis. So any cell, so also remember that meiosis is a cell division which is taking place in the sex organs like ovaries in the females and the testes in the male. Meiosis is the division which is not taking place in every organ of our body. It's only taking place in the sex organs. Unlike mitosis, which is taking place in all the organs of our body. So meiosis, before meiosis, there's an interphase, which means you can see like these are the chromosomes, and in the interphase they get duplicated. You can see how the chromosomes are becoming duplicated. And followed by interphase is meiosis 1. In meiosis 1, there is a separation of the chromosome. So this is a homologous pair. So you can see in meiosis 1, from one cell which has already undergone interphase, and in interphase there was duplication of DNA because there's an S phase. This cell enters into meiosis 1 and separation of chromosomes takes place. So this cell gets one chromosome, this cell gets another chromosome. This is happening in meiosis 1. And in meiosis 2, there is a separation of the sister chromatids. So here, two sets of chromosomes. For every pair, there are two sets of chromosomes. In meiosis 1, the number of chromosomes, they reduces to half. Here there were two chromosomes. Now this cell has one chromosome. This cell also has one chromosome. After that, after meiosis 1 is meiosis 2. And in meiosis 2, there is a separation of the sister chromatids takes place. So from this cell, two cells are going to be formed and each cell has one sister chromatid. Same thing here. So you can see here in meiosis 1, meiosis, it starts with one cell, one parent cell, and it ends up with 4 cells, and they are haploid. And these haploid cells, they are all unique. And I'm going to show you how these cells are unique. What happens during this process that it makes them, all of them, unique. Okay, so meiosis 1. So let's first of all talk about meiosis 1. Meiosis 1 has 4 phases: prophase 1, metaphase 1, anaphase 1, telophase 1, followed by cytokinesis. Cytokinesis, again, it actually initiates at anaphase. Okay, prophase 1. So something very important. So mostly most of the events which were taking place in mitosis, they overlap. So a lot of events which were taking place in mitosis, they also take place in meiosis. But there are some differences, and that's what we're going to talk about. So prophase 1 of meiosis 1 has that 3 important things which are taking place in mitosis. They will be also taking place in prophase 1 of meiosis 1, which includes nuclear membrane fragmentation, chromatin getting condensed and taking the shape of chromosomes, and the centrosomes moving apart from each other, and the spindle apparatus starts forming. So that will also take place in prophase 1 of meiosis 1. But something important and different takes place in prophase 1 of meiosis 1, which does not take place in the mitosis, and that event is called crossing over. Now, what is crossing over? And crossing over results in the formation of X-shaped regions called chiasmata, and they are the sites of crossovers. So let's go over here and talk about. So you can see here, so in the prophase 1 of meiosis 1, all the other events which are taking place in mitosis will be taking place. But in addition to that, few other things takes place, and that include, number 1, the pairing of chromosomes. The chromosomes, they start pairing up. Every chromosome goes and looks for its partner. Now you can see here, this pair is formed, this pair is formed, this pair is also formed. So in every pair, one chromosome is maternal chromosome, another chromosome is paternal chromosome. And you can see that these chromosomes, they are duplicated. Every chromosome has 2 sister chromatids, because this cell has already passed through interphase. And in interphase, there was S phase, the synthesis phase, where the duplication of DNA has taken place. So you can see here that these are the 2 arms of the chromosome, and these 2 blue arms, they're called sister chromatids. And in this red chromosome, these 2 arms are called sister chromatids. Okay. Now, in the pair, this red chromatid and this blue chromatid, these are called non-sister chromatids. Okay. These are called non-sister chromatids. And what happens is, these non-sister chromatids, so when the pairing takes place, the genetic exchange takes place between the non-sister chromatids. So which means that, and this event is called crossing over. And when these chromosomes, when these chromosomes, they go through the genetic recombination, or when these chromosomes, they basically physically exchange the genetic material, and then this X-shaped structures are formed. And they are called chiasmatas. So you can see here, this red chromatid is crossing over with the blue chromatid, and these chromatids, they are actually going to exchange the genetic material with each other, and that is going to result, these X-shaped structure called chiasmata, this event is called crossing over, and that results in the formation of recombinant chromosomes. So you can see, it's very clear here that this is a red chromosome, and it has some part of the blue chromosome. This is a blue chromosome, it has some part of the red chromosome. And why there's a formation of recombinant chromosomes? Because of the event which is taking place here, which is called crossing over. And this crossing over is responsible. This crossing over is responsible for the shuffling of the genes. So in all of us, we have one chromosome from our mother, one chromosome from our father. So during meiosis, what is happening is, these non-sister chromatids, they are physically exchanging the genetic material between each other, and this is causing the shuffling of genes between the paternal and the maternal chromosomes. And that is why we basically pass mixed characteristics to the next generation, because of this event. Because here the shuffling or the recombination of genes is taking place in the prophase 1 of the meiosis 1. Okay. Okay, so this is something very important which is taking place in prophase 1. Another important thing which is taking place, followed by prophase 1, is metaphase 1. In metaphase 1, in metaphase 1, unlike mitosis, in mitosis all the 46 chromosomes, they came in the center. But unlike mitosis, in the metaphase 1 of the meiosis 1, it is the pairs which come in the center. It's the pairs which come in the center. And you can see here, this is the maternal chromosome, this is the paternal chromosome, and these chromosomes are all recombinant chromosomes now. So this blue chromosome has some part of red chromosome, and red chromosome has some part of blue chromosome. So it's a pairs which come in the center. It's a pairs which come in the center. And in the anaphase, it is the separation of the chromosomes. It is not the separation of the chromatids, the way it was happening in mitosis. So in the anaphases, this is very, very important thing to know. In the anaphase 1 of meiosis 1, it is the separation of chromosomes. It is not the separation of chromatids. Okay, and then it is followed by telophase. So all the same things which were happening in mitosis, that the nuclear membrane starts coming back. So remember that on this side, which is going to form a new daughter cell, it will have 23 chromosomes. This side of the big cell, which is going to form a future daughter cell, is going to have 23 chromosomes. And do not forget, these 23 chromosomes on this side and 23 chromosomes on this side, they are duplicated chromosomes. They are duplicated. And again, cytokinesis, cytokinesis actually initiates in anaphase, and you can see how the cytoplasm is getting separated. So all those things which are happening in the telophase of mitosis, same things are happening here. Okay, the mitotic spindle is disappearing. Another important thing I would like to tell you, one thing here is, at the metaphase 1 of meiosis 1, you can see here the chromosome pairs are coming in the center. The chromosome pairs are coming in the center, and it is the separation of the chromosomes. Now, something very important to learn here is something which takes place here. It's called independent assortment of chromosomes. So what is happening here is, you can see here in this pair, the blue chromosome is on this side, red chromosome is on this side. So this side is going to form one cell in future, this side is going to form another cell in future. So this chromosome, so this is the alignment of this pair. So in this alignment, this red chromosome is going to go on this side, blue chromosome is going to go on this side, right? So how does this chromosome pair aligns? It is all independent of all these pairs. Okay, so this blue chromosome can be on this side, the red can be on this side. If that is the case, this cell is from this pair, this cell is going to get blue chromosome. This cell is going to form red chromosome. It's going to get red chromosome. And how are these chromosomes aligned? It's all independent of this. Okay, so the thing is, if suppose the orientation is opposite, red is on this side, blue is on this side, then this cell from this pair will get red chromosome, and this cell from this pair will get blue chromosome. And how these are aligned? It's totally independent. So which means that because of how this pair is aligned, or how this pair is aligned, or how this pair is aligned, there are multiple or million combinations possible. Million combination possibles, how gametes are formed, or what kind of gametes are formed. And this process is called independent assortment of chromosomes. Okay, so these are some of the important events taking place here. Number one, crossing over. Second, independent assortment of chromosomes. That results in the formation of different kinds of gametes. And that is responsible, as I said, in meiosis, from one parent cell, 4 daughter cells are formed. Number one, they are haploid. Now we understand why they're haploid, because there was reduction in the number of chromosomes. And also, they're all unique. Why they're unique? Because of, number one, because of crossing over, and number two, because of independent assortment of chromosomes. So these two events are very big contributors of the uniqueness of the gametes. Okay, and then so it means that from one parent cell, two daughter cells are formed, and those two daughter cells, they are haploid. And then meiosis 1 is followed by meiosis 2, and it also has stages like prophase 2, metaphase 2, anaphase 2, and telophase 2. And meiosis 2 is exactly like mitosis. So in prophase 2, there is no crossing over, nothing. And in the metaphase 2, it is actually, so after meiosis 1, there's a formation of two daughter cells. And these two daughter cells, each cell will undergo meiosis 2. So this is one cell, this is second cell, and you can see in metaphase 2 of meiosis 2, all the 23 chromosomes, they come in the center. So this is one cell, this is second cell, all the 23 chromosomes come in the center. So here is one difference between, so we say meiosis 2 and mitosis are exactly same, but there is one difference. Then in metaphase 2 of meiosis 2, there are 23 chromosomes. But in the metaphase of mitosis, there were 46 chromosomes. And in the anaphase 2, it is actually the separation of the sister chromatids. So in anaphase 2, it is the separation of sister chromatids, just the way it is in the anaphase of mitosis. And followed by telophase and the cytokinesis. And so this cell will form two cells, this cell will also form two cells. So in total, from one cell, 4 haploid unique gametes are formed. So we talked about what is crossing over and what is synapses. So basically it is showing you in more details that these are sister chromatids. So one blue chromatid and one red chromatid, they are non-sister chromatids, and you can see how physically they are exchanging genes here. So this is called genetic recombination, or we can say gene reshuffling is taking place, or gene shuffling is taking place. And these X-shaped structures which are formed, they're called chiasmatas. And you can see how these chromatids, they are actually exchanging genes with each other. This is what is happening in the during the crossing over. So you can see here how this is a blue chromatid, how this is a red chromatid, how they are exchanging genes. Okay, this is just a comparison of mitosis and meiosis. So you can see here in the metaphase, all the 46 chromosomes coming in the center, and in the meiosis 1, the metaphase 1 of meiosis 1, it is the pairs which come in the center. It's the pairs which come in the center. And then, so in the mitosis, it is the separation of the sister chromatids. In the meiosis 1, it is the separation of chromosomes. And from one cell, two cells are formed. From one cell, two cells are formed, and they have half number of chromosomes. And the chromosomes are duplicated in meiosis 2. In meiosis 2, there is a separation of the chromatids. So genetic variation produced in the sexual life cycle contributes to evolution. Yes, because genetic variation is actually very important. So mutations are also responsible for genetic variation. So also during this process, so reshuffling of alleles during sexual reproduction produces genetic variation. So we just saw during the crossing over process how the reshuffling of the genes takes place. And I hope you, the word allele basically means, let me explain you from here. Let's go back. Here is one term called allele. Okay, let me explain you from here and then I will go back. Like this is one chromosome. This is a duplicated chromosome. This is a second chromosome. Okay. Okay, I think it, let me go back here. Allele word basically means so basically it should be explained from here actually. Let me go to the karyotype and I can explain you from there. Here. So basically these are homologous pairs, right? This is pair number 1, pair number 2, pair number 3. So in every pair, one chromosome is from father, another chromosome is from the mother. One is a paternal chromosome, another is a maternal chromosome. So if suppose here we have a gene which is responsible for skin color, or any character we can consider, any character. So if there is one gene here which is responsible for one character on the maternal chromosome, at the same locus, we have the same gene which is coding for the same character on the paternal chromosome. So this gene and this gene, so this gene and this gene, these are two versions of the same gene which is coding for the same character. These are the two versions of the same gene coding for the same character, and these are called alleles. This is what is the meaning of the allele. Okay, so now we are almost at the last part of this chapter, in which we are going to talk about what is responsible for the genetic variation. So there are basically three mechanisms which contribute to genetic variation. Number one, independent assortment of chromosomes, which we just talked about, crossing over, and third is the random fertilization. So during the process of meiosis, gametes are formed, sperms are formed, then eggs are formed, and they're all haploid, they're all unique. And then after that there is a process of fertilization. So the sperm goes and fertilizes the egg, and which sperm is fertilizing which egg, that is also a random process. So that is why that is called random fertilization, and that is also responsible for genetic variation. So independent assortment of chromosomes, we already discussed what does that mean, and because of that there are around million combinations of gametes possible. So now you can see here, this is one example to understand independent assortment. Going back here. So you can see here, this is possibility number 1. So in possibility number 1, both the blue chromosomes which are the paternal chromosomes are on one side, and the maternal chromosomes are one side. So suppose this is, so we are just talking, so in humans we have 23 pairs, okay? We are just talking in terms of only 2 pairs of chromosomes. So if both the paternal chromosomes are on this side, maternal chromosomes on this side, what will happen? This cell will divide, okay? This cell will undergo meiosis 1, and it will result in the formation of 2 cells. So in this orientation, this cell will get both paternal chromosomes, and this cell will get both maternal chromosomes, and they are duplicated. And then from one cell, 2 cells are formed, and then these 2 cells will further undergo meiosis 2. And from this cell, 2 cells will be formed. From this cell also 2 cells will be formed. So in total, 4 haploid cells. Now from this cell, because it has both the chromosomes, paternal chromosomes, in meiosis 2, it will be the separation of the chromatids. So from this cell, when 2 cells will be formed, so one cell will get this chromatid and this chromatid, one big blue chromosome chromatid and one small blue chromatid, and the same thing here, and same thing here for this cell. When this cell will divide into 2 cells, one cell will get this one red chromatid, big red chromatid, and second small chromatid. Then this second cell, one big chromatid, another second small chromatid. This is the combination possible if this is the orientation. But suppose if the orientation is like this, blue chromosome on this side, but for the second pair, red chromosome is on this side. So you can see that in meiosis 1, 2 cells will be formed. It will have one blue chromosome, one red chromosome. This cell will have red chromosome, blue chromosome. And when they undergo meiosis II, these are the kind of gametes possible. And you can see how different they are from these gametes. This is a case of 2 chromosome pairs. In humans, we have 23 pairs of chromosomes. How one chromosome pair is aligned, it is totally independent of the other pairs. So that is why there are millions of combinations of gametes possible. So this is one of the very, very important event or mechanism. It's a kind of a mechanism which is responsible for genetic variation. Crossing over, we talked about what crossing over is. It is basically, you can see here that how these non-sister chromatids, they are physically exchanging genes, and how recombinant chromosomes are being formed. And then you can see this is a blue chromatid with red part, and this is a red chromatid with blue part, and how in the anaphase 2, so this is anaphase 1, where the separation of the chromosomes is taking place, and in the anaphase 2 it will be the separation of chromatids, and that will result in the formation of daughter cells. And random fertilization, now which sperm, so we just talked about the process, how eggs and sperms are formed, and who finds which partner, it's all random. Which sperm is going to fertilize which egg, it's all random. So that is the third mechanism which is responsible for the genetic variation. So that's all in this chapter.